Welcome, everyone, to our Defeat Duchenne Canada webinar series. I'm very pleased to have Solid Biosciences with us here today to share an update on their clinical trials. As most of you know, Defeat Duchenne Canada is the country's only national charity dedicated to ending Duchenne muscular dystrophy. Our goal is to provide leadership in research and advocacy while supporting all Canadians with Duchenne. Our vision is a future without Duchenne. My name is Judy Wiles. I'm the Director of Research and Advocacy. Just here to quickly introduce our 4 strategic pillars: research, advocacy, education support, and community engagement. We are very pleased to welcome Solid Biosciences here to give us an update, and I'm going to pass it over to them to give their update.
That's great. Thank you so much, Judy, and the entire team at Defeat Duchenne Canada for hosting us today. I'm joined by my colleagues, Pat Gonzalez and Matt Harmelink from our clinical team. Sorry, next slide, Pat. Thank you. That's great. That's us. And then next slide, please. As a publicly traded company, we may be making some forward-looking statements. Next slide. We circulated a letter to the Duchenne community last week with an update on INSPIRE Duchenne, Solid's AAV microdystrophin gene therapy clinical program testing our next generation construct, SGT-003. As we are opening our first clinical site in Canada at SickKids Hospital in Toronto, we are thrilled to have the opportunity to speak directly to you all and do our very best to answer your questions. My husband and I founded Solid in 2013 following our son's diagnosis. We wanted to do everything possible to improve the lives of all boys and young men living with Duchenne. Over the past decade, we've grown significantly as a company in clinical research and development and manufacturing expertise, all in close partnership and informed by the Duchenne community. Utilizing these advancements, we have recently expanded to additional related neuromuscular and cardiac indications. Our plan today is to take you through the science of SGT-003 and why we're excited about the potential of this program. We will then share an overview of the clinical study, including recent expansion of eligibility criteria and site locations. And of course, we're happy to take your questions after the presentation. Please feel free to drop any questions you may have in that Q&A box, and we'll do our best to get through everything. Thanks again for joining today, and I'll now hand it over to my colleague, Pat Gonzalez.
Thank you, Annie. Thank you, Defeat Duchenne Canada, and everyone here for joining for us for today. As Annie said, we're really excited to really give the first introduction of this program to Canada as we are now moving forward and activating our first clinical trial site. So what I wanted to do today to start is really give the overall overview of what is SGT-003, what are the main pieces of this approach that we're taking to treat Duchenne muscular dystrophy, and then highlight a lot of the data that we've generated that pushed us into initiating this clinical trial, which is now active and ongoing. And Matt will take us through that in the second half of the talk here. So overall, SGT-003 is an AAV-based gene therapy. And this means that as a gene therapy, just as a quick introduction and overview here, we're looking to really deliver a transgene to be able to restore a functionality that's missing. And so in Duchenne, we know that because of a mutation in the dystrophin gene, we have the loss of a functional dystrophin protein. And that then causes that muscle instability and breakdown over time. With an AAV-based gene therapy approach, the goal is to use here what is our delivery vehicle of an AAV capsid. So really taking advantage of the properties of this to be able to then use a systemic administration to get something throughout the body. We use a promoter that can then constrain that expression to specific tissues or cell types. And so for Duchenne specifically, we want to make sure that we are active in really all of the target tissues throughout the body, which are skeletal and cardiac muscle, most importantly, and very much inclusive of the diaphragm muscle as well. As we're looking at this approach, we need to be able to deliver something through this AAV. And the transgene specifically here is going to be our cargo that we're delivering. And I'll go through a little bit of the details of what this specific transgene is. And because it's a microdystrophin and it is a treatment for Duchenne, we're looking at pieces of a full-length dystrophin that we're then restoring here to get that functionality back. So altogether, SGT-003, we call a next generation AAV microdystrophin gene therapy. And the reason that we call it a next generation are because of a couple of key components that really have been optimized over time and that we believe, based on the data that we've seen so far, could be meaningfully differentiated for the treatment of Duchenne. So I talked about the transgene that we deliver through this approach. And for this program, we are delivering a unique microdystrophin transgene. When we think of different ways that we can look to restore dystrophin functionality, if we're using an AAV-based approach, one of the limitations is the actual size of what we can put into that delivery vehicle, into that capsid. And so as microdystrophin gene therapies were thought of, we needed to be able to choose different regions of the full-length dystrophin to be able to put into this delivery vehicle to then be able to get to our target tissues. Our specific construct, and I'll go through this a little bit more in the next slide, is inclusive of what's called the nNOS binding domain. And so we know that dystrophin needs to function from a structural side, and that it has to link the muscle so that it can then withstand contraction and relaxation, but it also has other functions as well. And one of these being the ability to recruit this domain, through this domain, nitric oxide production at the muscle membrane. We'll go through why that's important in a second. As we're looking at a next generation approach, really the key to everything here is how we're able to deliver this transgene to our target tissues. And so what we did as a company was really go back to the lab and see if we're looking at these different capsids, these different AAVs that could be used for the purposes of getting our transgene to tissues, is there some way that we can improve upon what has already existed, improve upon the first generation or the naturally occurring capsids? And so this is where this specific capsid called AAV SLB101 came through. Really through many screens that we did in the non-clinical side, looking at in vitro lines, looking at animal models to say, is there a way that we can get more of our delivery to our tissues? And specifically, as you can see here on the bottom, we were able to achieve that in non-human primate studies, looking at multiple fold higher levels of expression in muscle tissues, including the heart and the quadriceps. And we'll go through these again. At the same time, because we know we're administering high doses, our goal is always to be able to see how can we reduce anything that's going to off targets. If you're giving a systemic dose, that's going to go throughout the body, and a lot of that will still go to places that you don't need it to necessarily. And so while our promoter is going to restrict the expression to the tissues we still want to overall limit the amount that's getting to other tissues. And so one of the components of selecting this capsid was also due to the profile where we could see that by increasing the amount that's getting to muscle, we also decrease the amount that's getting to the liver. And so more to muscle is a better thing, less to liver is a better thing as well. And then lastly, as we came together to really put this program together and be able to make enough for patients, we really wanted to see what can we do from a manufacturing side to maintain that high amount that we need to make to really treat all patients with these high systemic doses, and also maintain a purity profile that could be beneficial as well. And so again, because we're administering high doses, anything that we can do to reduce the total amount of particles, which we usually characterize as these two different pieces here, either full particles, which contain the transgene, or empty particles, which don't. If we can reduce those empty particles that come along for the ride when you're making this product, then we're going to have less total viral load that's getting to patients and could potentially have a beneficial effect on safety. In addition, what we saw and what's highlighted here is that when you improve those full particles, so the ones that have that transgene, we also see that you can improve your expression levels as well. And so together, bringing together what we have as a transgene, this capsid and process really came to make SGT-003. And we'll go into these details a little bit more as well. And so I think all of us here know really what is dystrophin. And so from a very high level, it is mainly a structural protein that the first thing you think of for dystrophin is that in a muscle, you need dystrophin at the membrane to be able to join the intracellular side to the extracellular side. So really to make that physical connection so that when you are contracting and relaxing your muscle, it has the ability to withstand that process and it won't break down over time. We know that when this protein is missing, that's really the effect, is that now you don't have the ability to stabilize that muscle fiber. And so you do have that progressive degeneration, regeneration over time that then leads to the loss of overall muscle and strength. So in a microdystrophin approach, as I mentioned, we do have a capacity constraint that we need to work with. And so each of the different pieces that we're looking at need to be rationally put into this microdystrophin to understand that each is going to confer a specific functionality that we need to restore as much of that full-length dystrophin function as possible. And so this is really work that's been originated from initial Becker studies, understanding what are the functional pieces of a dystrophin that are critical to be able to then, without the full-length protein, actually restore some level of functionality. And from there came really seminal works from Dongsheng Duan and Jeff Chamberlain that showed us that there are these critical domains that are required for things like that structural function, and then there are other domains that are required for things like molecular function, and that's this nNOS binding domain. And so when we think of the nNOS binding domain and what does this do, it's really something that becomes most important when you are in the setting of energy demand. And so when your muscle is going through exercise, when you're using it, you are contracting and relaxing, you know that that's going to require more energy to get to the muscle. The way that our muscles normally do this is by then activating something called nitric oxide, which is able to increase the blood flow to the muscle in this context. And so when dystrophin or a microdystrophin even that doesn't have this binding domain is absent, we aren't able to actually get nitric oxide to the muscle membrane and we can't get that blood flow. And so by restoring this specific form of microdystrophin that has this additional functionality, we have the potential to be able to actually include this additional potential prevention of something what's called ischemic induced injury. And so ischemic, if you think of basically not getting oxygen to the muscle as necessary, so now that can cause an injury. If we can be able to prevent that, that could have a potentially meaningful effect as well. And this has been a core reason for why we focused on using this microdystrophin for our SGT-003 gene therapy approach. And so a lot of this matters in how much can you actually get to the muscle ultimately. And so because this is a systemic disease, we need to be able to treat as many muscles as possible. Using an AAV-based approach, this will then be a single systemic administration that we use both non-clinically as well as clinically. And so what we wanted to do was look at those first generation capsids that existed and say, can we actually find a way to improve upon what has existed? And so back in the lab, really what was looked at are different ways to modify the capsid or the coding that we have on this delivery vehicle. And so if you think of the actual coding of the capsid as basically the GPS that's going to tell the capsid where does it need to go into the body, if we can try to refine those instructions and those directions a bit, then we could potentially get more to where we need it to go and less to where we don't need it to go. And that's essentially what we did through multiple rounds of screening, looking at different modifications to naturally occurring capsids to use this rational approach to say, if we can target X on a muscle fiber, that could potentially get us to a point where more is getting to where it needs to be. And so that's what really came to be SLB 101, this serotype of AAV. And you can look at the data here on the right side, that when we use this in a mouse study, we can see that we do get multiple fold higher levels of distribution of this capsid and this capsid containing our transgene to our target tissues. So the quadriceps here as a representative skeletal muscle, and then the heart here as well, both very important and critical tissues to be able to get good expression to ultimately. With that, and as I showed in the previous slide, we also saw that while we targeted muscle better, At the same time, we also received a profile that resulted in less going to the liver. And so again, while more is better for our target tissues like quadriceps and heart, less is better for the liver because we have less that's going to just be a viral load that's going somewhere where we don't necessarily need it to be expressed. So ultimately, these data and others that we use to scale up have really made us excited about the potential for this capsid. And so one of the first studies that we did after we looked at in vitro settings and then also our early small animal studies were really to move quickly into what are called non-human primate studies. So wanting to move into monkey studies that could start to say, if we're looking at larger animals, if we're looking at something that's a bit closer to a human, how does this translate? Can we actually get that similar response that we saw from the small animals? And so what we were able to show, and here is using our AAV SLB101 compared to an AAV9, is that that same pattern that we saw in the mouse studies did hold. And so we could see here again in skeletal muscle, multiple fold higher biodistribution, the same in cardiac muscle as well. And again, about half of the amount getting to the liver. And so each of these really following the same pattern that we saw from our small animal studies and giving us hope that this is something that could translate. We continued on to also look at other capsids that could be those first generation approaches. And so wanting to really understand that if we're going to look at a next generation approach, we want to compare against something that's being used widely right now. And so our major study that we ran here was comparing our AAV SLB101 to AAV RH74. And so what we did was actually package a full therapeutic transgene, so our microdystrophin with a muscle-specific promoter, into each one of these two capsids. We administered them to separate animals at equivalent doses, and these are the results that we saw. In the heart, we had approximately 4 times higher levels of microdystrophin expression using the AAV SLB101 capsid over RH74. And in the quadriceps, so again, another representative skeletal muscle, we saw up to 6 times higher expression of microdystrophin expression. And so these results really came together to say that there are potential for this capsid to have a meaningfully differentiated profile from the first generation approaches, either from AAV9 or RH74 that have been looked at to date. So really from there, what we wanted to do was quickly move into IND-enabling non-clinical studies. So to put together the package that we needed for regulators to be able to get to allow us to move into our first clinical trials. And so I'll highlight a few of the pieces of data here before getting into the clinical trial design that we actually are moving forward with now. And so as I showed previously, what we can see is overall our AAV gene therapy approach is aiming to deliver this microdystrophin transgene. So we need to get into our muscle, we need to be expressed, and then we do want to show that it's functional as well. And so one of the first things that we can look at in terms of functionality is whether it's able to actually localize to the muscle membrane. And because it has to link to the muscle membrane to have that structural stability, this is a key initial piece of looking at whether a microdystrophin is functional. So what we could see here in a dose ranging study is that we do see dose-dependent increases in the microdystrophin levels, and these are correctly localizing to the muscle fiber membrane in each one of these muscles. With that, as I mentioned, we do have this unique functionality of this SGT-003 microdystrophin to also recruit nNOS. So we looked at nNOS activity as well in these tissues. And so here in a vehicle, so this is an MDX untreated animal, so a dystrophin negative animal model of Duchenne, we don't have that nNOS activity at the muscle membrane. But just as with microdystrophin, when we increase the amount of microdystrophin that's expressed by dosing with SGT-003, we see that we have a corresponding restoration of nNOS activity to the muscle membrane in each one of these tissues. So when we take that from a quantitative perspective, we also want to look to see, are we getting enough throughout that entire muscle? Are we distributing our microdystrophin as much as possible? And thinking about this in terms of protecting muscle, it's always important to not just have a small amount of a high amount of expression in a small amount of the tissue. You want to be able to target as much of the tissue as possible because ultimately all of it's going to come together for functionality. And so what we can see is that even at our low doses tested here, so as low as 3E13 in this specific study, we start to see quite high levels of microdystrophin expression even at day 4 post-dosing. And then once we get to points where we have understood really that this is now where you start to more so stabilize or reach higher levels of expression, we could see that at day 29, so a month out post gene therapy administration, we have approximately 100% positive fibers at each of these dose levels and in each one of these tissues that we're looking at. So the diaphragm, quadriceps, gastrocnemius. So looking at really core muscles, limb muscles, and then the heart as well to make sure that we are getting that expression and at these levels which are actually getting to entire muscles. And so as we looked at that initial molecular functionality, we also want to see how does that work from a physiological sense? Are we able to actually show that microdystrophin results in a different type of function actually from what you can see from an activity side? And so we looked at a couple of different methods here. The first on the left side looking at treadmill exhaustion. And this is a way to say that do you have the muscle endurance to actually perform this task better than without having a treatment? And the answer was yes. Even at that lowest dose level tested, we saw a significant improvement. And on the right side here, what we wanted to look at was actual muscle force. So looking at the EDL, which is a lower leg muscle, what we can see is that when we look at how much force can this muscle actually produce to really say, are we stronger because of this microdystrophin treatment? We could see that same pattern held true as well. So even the lowest dose tested had a significant restoration of function and back to wild type levels, which is really interesting here. So I think there's always been a lot of questions on kind of what's the potential for a microdystrophin to restore functionality. At least from the assessments here, we know that we have that potential to restore this strength, which could be really meaningful overall for a therapeutic approach. In addition, we look at things like biomarkers to really say, when we're looking at this in the clinic and we need to see early signs of changes, are there some other circulating markers that might help us tell that story? And so I think one thing that we all understand is actually serum CK, so creatine kinase, which gets leaked from the muscles in Duchenne. And that's really just because the membrane is unstable. And so when you have that unstable membrane, you have leakage that comes out, but that then goes into the circulation. And that we were able to see is that we also have that significant decrease near the wild type levels here when we have treatment. And again, even at that lowest dose level, getting to that point. That then held true with other markers that we looked at. This is one more novel marker that we're exploring called serum titin. And so again, looking for this to be in the circulation as a serum titin fragment to again correspond with the CK and say, are we leaking less into the circulation? And this then had the same pattern and even at the lowest dose level tested, again, a significant reduction down. Here in AST as well, seeing that dose response more so, but again, all of our biomarkers corresponding with what we saw from our functional tests and then molecular tests as well. So really this data set together, put together our package, which we initially brought to the FDA in the US for the advancement of this program into clinical trials. We believe that SGT-003 does represent a next generation therapy because of these pieces that we just mentioned, the unique microdystrophin, the novel capsid that's being used that's rationally designed and does seem to have a better profile for getting to our target tissues and less to off-target tissues. And overall, the data that we were able to generate from a functional side made us very excited to be able to push this forward. And so I'll hand it over to my colleague Matt here to introduce you and really everyone to the INSPIRE Duchenne study and why we're so excited to talk to you today about how we're advancing this into Canada as well. Matt?
Thank you, Pat. Really excited to talk about the trial. So if we can go to the next slide. So INSPIRE Duchenne is a phase 1/2 clinical trial to evaluate SGT-003. It's being conducted in the US, Canada, and Europe, and it is a single dose level open label study, meaning that there is no placebo arm. Everybody who gets into the study will be dosed. It's being done in ambulant patients with Duchenne's, and the prophylactics is an increase in prednisone from the baseline immunosuppression. So your steroids are increased. The trial design is made to be focused on safety because it is a phase 1/2 clinical trial. So the key endpoints are safety and evaluation at day 360, microdystrophin expression at day 90, and then the time to rise at day 540. Now, there are 2 cohorts in the study. One is age 4 to under 7. The other is cohort 7 to under 12. So exclusionary of 12-year-olds. We did that just to make sure we include the 11-year-olds clearly. And there is a weight limit of equal to or under 50 kilograms. Both cohorts are open and actively enrolling with a goal target enrollment of 43 patients. Next slide. The key eligibility criteria for the trial, as mentioned before, we are looking at ages between the age of 4 and under 12 for the different cohorts. There is a DMD mutation exclusion for anybody who has a deletion between exons 1 and 11 or 42 to 45 inclusively. For functional inclusion criteria, cohort 1 does not have any, but cohort 2 does have some criteria around time to rise and the 10-meter walk-run test as well. All patients must be negative for AAV antibodies, and they cannot have been on any prior gene therapy treatment, whether investigational or commercial. For exon skipping drugs, vamorolone, givinostat, there has to be at least a 12-week washout period prior to entering the study. For those who are on vamorolone, they would have to switch over to another standard of care, either deflazacort or prednisone steroid at that time. Next slide. Because this is a study focused on safety, for the first 30 days, everybody has very frequent visits to the hospital, getting checkups and blood work. At that point, it does start to get weaned less frequently, but it is a long-term 5-year post-administration follow-up study that's required by regulators. For efficacy measures, there will be 3 total muscle biopsy performed during the study. The recommendation for the study is to use a needle biopsy technique. Those sites can have preferences of using an open biopsy if they feel the need to. These biopsies will be done at screening, day 90 and day 360, and they're being used for evaluation of microdystrophin expression. There is also other testing being done to measure motor function, pulmonary function, and other patient-reported outcomes that is throughout the course of the study. Some of these are in-clinic tests and questionnaires, but there's also a component that's at-home video and activity monitoring as well. Next slide. Now, to date, we have achieved some milestones already. We had our first clinical site activated in the second quarter of 2024, as well as the first patient dosed at that time. To date, by the end of quarter 3, there were 3 patients dosed with SGT-003, and no significant serious adverse reported events have been reported at this time. We initially also, for those who may have followed the program, there was a smaller safety study that was expanded in the third quarter of this year to have a much larger study to evaluate some of the efficacy targeting that we could do with more patients being enrolled. So upcoming events, well, we currently have active sites that are screening and enrolling patients. There are some upcoming active sites that are identifying patients for potential participation, and we hope that in Q1 of 2025, we'll have the 90-day data from the first 3 participants that will be able to be shared. Next slide. There's also clinical site expansion underway. In the US, we have multiple additional sites that are in study startup, with new activations hope to be starting as early as this quarter and continuing forward. In addition, as already mentioned, SickKids is in the process of a study startup as well, and so we're excited to bring them into the study with expected activation in the fourth quarter of this year right now, so anytime. Additionally, there will be some sites in Europe that are being done in parallel with regulatory activities, and those clinical trial applications have been submitted with the expectation of starting in the first half of next year. Next slide. These are the current sites that we have either active or very close to activation right now. So for geographies as people are looking, and I will leave this just for one moment, but on the next slide, there is information actually to how to contact sites if you're interested in hearing more. Next slide. And here are actually the sites that are either active or active very shortly, including contact information for interested families who want to learn more about the study. On the bottom, you'll note we have our QR code that will navigate to the study posting on clinicaltrials.gov that is kept up to date. If there are any questions or concerns, please feel free to add them into the Q&A at this time. We're happy to take questions.
We do actually already have one question. This is Judy, and I'm just speaking on behalf of one of our participants. Anna comments, one of the exclusions for the clinical trial is exons 42 to 45. Does it mean if a child has a deletion from 45 to 52, he will be excluded from SGT-003?
I'll get off mute fastest. So Yeah, so currently, really out of an abundance of caution, and I'm always happy to go into the details on really that specific exclusion criteria. Based on the way that we're delivering a microdystrophin that has not been seen by the body previously, what we're looking to do is be conservative from an exclusionary standpoint and say that if we are administering something that is part of a deletion that hasn't been seen, that we don't want to include that patient at this exact point as we learn more. So a 45 mutation that starts as deletion would be excluded right now. And so that would be similar to 1 to 11. Any piece of that that is a deletion specific mutation would be excluded, and the same thing as 42 to 45. And this is something that I'll also just say, I think we're hoping to learn more with the field, especially as we understand what are the immunogenic regions. Are there ways that we could be able to change these criteria over time? But again, as this is our first in human study here, really starting out of an abundance of caution as we look to then proceed.
Thank you. Are there any other questions? Please feel free to type them into the Q&A section. We can also see your chats if you want to put in there. If you want it to be more anonymous, if you put it in the Q&A, only the panelists can see the questions. But don't be shy, people. We're fine either way. You had mentioned that that SickKids is opening up within this quarter, which is great, which means probably within the next 6 weeks at the most, right? Are there many Canadians who are going to your US sites already with SickKids not quite being up yet?
Yeah, I'm glad you asked that. I think what's important to know is that, especially based on proximity, and that's why we do want to highlight every site here. Multiple of these sites in the U.S. are also accepting Canadian patients, and so absolutely, whether it's SickKids or whether it's another location that's basically works best for a family, want to make sure that they have that opportunity to be able to reach out. So the answer is yes, and there are others who are still looking at each of these sites. To the point about SickKids right now, we are right around the corner from officially activating, so we are
Right.
hoping that that is coming up very, very shortly. Wanted to make sure to get that information out today, nonetheless, just to make sure that everyone is ready for that as well.
Wonderful. That's very exciting. We're glad to hear it. There are some questions coming in on the Q&A. So the age criteria, maybe you could back up to the slide about your inclusion criteria about age, just so people can take a quick peek at that if you don't mind.
Sure.
So someone was just asking about it. So it looks for cohort 1, 4 to less than 7, and then for cohort 2, 7 to less than 12. Do you have any intentions of moving into the adult age? And if so, where would you see that maybe coming up later?
So certainly there's been a lot of discussion about older patients. At this point, as Pat had already mentioned, we are very much focused on the safety of this current cohort that we're looking at. And that was very carefully selected based off of criteria for disease basis and other things like that. As we move forward, that is going to be part of the discussion that we'll be having.
Sounds great. Is there a cap for how many Canadian participants you can have?
Currently, we've not set any caps for sites. It's a competitive enrollment process.
Right. Someone is asking when you started your dosing. I believe that was covered on one of your slides, so it might be a backtrack situation.
Yep, sure. So we did start dosing right around the halfway mark of this year. So right at the end of Q2, and we had progressed through then for our next participants dosing. So the way that we had started this study was really conservatively wanting to make sure that we're monitoring each one of our patients very closely, looking at safety. And then so from Q2 through the end of Q3 is where we had our first 3 participants that were dosed with that spacing that we required so that we can all look at things and make sure that we're comfortable to proceed.
Great. Questions are coming in as we go. I think we've addressed all that are out there. Just checking to see if there's any in chat. coming
And
up.
maybe while we're on this slide too, just wanted to also
Please.
highlight the point about prior treatments as well. I think one thing as we think about as how we started the study back earlier in the year compared to now, a change that we wanted to make sure to make is as new therapies are being looked at, have the potential for approval in different geographies, we don't want to exclude patients just because they have had something before or have potentially been on an investigational study before. So especially as we look at exon skipping, vamorolone, givinostat, these are things where we require now this washout period, but patients would not be overall excluded just because they have had them in the period. So just wanted to point that out because it's a question that usually comes around, and we were very cognizant of the changing landscape and wanted to be mindful of that to allow patients to be included, but wanted to have this washout period just to reduce the variability as we don't know how one treatment works with the other yet.
That's great. Thank you for focusing on that. We're being asked if people are able to start the trial in the US and transfer to the Canadian site.
So at this time, our ask is that where patients start the trial, especially through the first 540 days, that they stay at the same site. We do understand that this is a 5-year trial and that things can happen, life happens. And so post that day 540, I think there would be a discussion with the site and then where the patient lives, things like that as they talk about moving sites.
Great. I believe you covered this, but unfortunately, I can't remember off the top of my head. How many people are you enrolling?
So the goal is to have 43 patients in the study.
Great. Sounds good. We're caught up on questions. Just a reminder to anyone who has questions, please feel free to put it in the Q&A, or you can also ask it in chat, whichever is easiest for you. This has been very helpful, and it's very exciting that you're moving into Canada. I'm really happy to see that, and I know there'll be a lot of people that also are. Do you have any plans in the future for any other Canadian sites, potentially maybe in the Western provinces by any chance? We're hitting you up before you even get Toronto up and running. We're asking and hitting you up for the West Coast.
I'll go straight to the map because we know that the Canada is obviously very large. Yeah, I think this is something that we're looking at right now very closely. We're starting to have discussions with more investigators, both for this study as well for potential studies in the future. So are very much looking at additional locations in Canada as well.
Great. Wonderful. Does anybody else, do any of our participants have any questions? Oh, there's some more coming in. Is there a requirement for a base treatment? So for example, do you have to be on steroids for a certain amount of time? And I know that we just talked about that you could have been on vamorolone or whatever and just needed a washout period, but do you have to be on steroids to participate?
You do have to be on steroids. And thank you, Pat, for bringing this up. The very last point here. So you have to be on a stable daily oral steroid regime of either prednisone or deflazacort for equal to or greater than 12 weeks.
But that's reasonable to be able to achieve that and then enroll, I would imagine, as long as you're still enrolling patients. So that's good news too. Are there any other questions for anyone? I think that's all we have right now. I'll just keep an eye open if there was anything else you wanted to cover off.
Yeah, I think maybe one thing to highlight is just as we kind of look through all of that non-clinical data and really the support for where we thought we could be meaningfully coming into patients with a therapeutic dose. This is the one thing I want to kind of highlight is just our dose level that we selected, which is 1E14. It doesn't always mean so much to everyone, but it was kind of right in the middle of the range of doses that we had evaluated. And we were really excited to see that even at lower doses in our smaller animal models, we did see really significant results and we were able to restore function. But as we came into this study, what we really wanted to push for was, can we see with acknowledging that this is a single dose treatment at this time, that redosing is not something that is currently possible. Can we see a meaningful effect and potentially that optimal level from this single dose? And so that's really the rationale for going in with this dose level, is hoping that if we're able to see anything close to what we saw from the non-clinical, this could be meaningfully different. So overall, always just want to show that. And of course, if there's any other questions for non-clinical data or anything else, we're always happy to answer that as well.
We do have one more. If you have any potential sites in Europe at this point.
Yes. So currently we are moving towards Europe as well. And so we did announce that our hope is really to have multiple site activations actually within the first half of 2025. So more to come on these as we actually have each of these regulatory submissions moving through, and then also site activities coming through over that next quarter or two.
That sounds good. There's one other question. Will you be using SV95C? And if so, is there a specific device you'll be using?
Yeah, that's a great question. So as we looked at all of the different outcomes that we can look at here, and I'll just quickly go over to all the different things that are done. So we don't highlight everything, but I think that's one of the more important ones that we want to make sure that we're incorporating as much as possible. So we're looking to be able to incorporate that as well, hoping to get that through into Canada for this study to use the side device as well, which has been used especially with the EMA towards a primary endpoint qualification, which is just something that happened more recently within the last year. So we think it can be a meaningful support of evidence. And so I'm really happy that was brought up, because I think one of the most difficult things is being able to get objective results from a study. But when you look at something like SV95C, which for those who don't know is stride velocity, but the 95th centile. So basically what your peak performance can be. It's something you can't really control on a day-to-day, but you can reduce a lot of variability. So it's a very objective measurement. It's something that we're absolutely looking at and hopeful that it can be able to support a lot of these in-clinic assessments.
Great, and thank you for the explanation of it as well. Do we have any other questions? I think right now we have come to the end of the questions, unless going once for anyone else who has anything they needed answered. I think that's it at this point. Did you have any additional slides you wanted to go through?
I'll just put this one back out there just to
Perfect.
hold on for a second. I think, as we mentioned, we're really excited to be able to move into Canada with this new site that's coming online very soon. We're excited to be expanding the study in general. And just based on what we've seen from the first few participants who have been enrolled, have been happy to see the positive safety profile to date. And so with that, we'll continue to be updating the community with everything that keeps moving forward. I know from a communication standpoint, especially direct from patient advocacy, and I'll tag in Annie here as well. We always want to make sure to maintain that kind of at our core, a direct connection to patients, families, and the community.
Great.
Just to add, we'll continue to keep the community updated, and to note that there is, again, the QR code. So if anybody does want to take a picture of that, it takes viewers directly to clinicaltrials.gov with all of the information that is publicly available for the clinical trial.
Wonderful, thank you. We have one question about if there's any chance of reducing to 4-year-old inclusions.
So currently the way the trial is written is it is a strict criteria as 4 years. Obviously, as we look at the field of Duchenne's in general, there is interest, as we mentioned before, about the older boys and adults, but also going on the other direction as well. But for the trial currently, you'd have to be 4 years of age at the time of screening.
Wonderful, thank you. And we have a few more coming in. To the person asking, yes, the webinar is being recorded, so it will be accessible later. And if there's anything else anyone would like to cover off, I think it's probably going to the last moments. Is there anything you all would like to cover off as well?
This was great. Thank you guys so much. Thank you for hosting us, Judy, Defeat Duchenne Canada. We're super grateful for your time and your interest. And if there's any questions, obviously do feel free to reach out to any of the sites or directly to us here at Solid.
Thank you so much for being here, giving us the update and all the exciting news about SickKids opening up very soon. We'll definitely be watching closely. Thank you so much, everyone, for coming into this. And the webinar will be available shortly. A recorded version, we'll send that out on the socials. So you can please feel free to look for us there.